So my name is Diogo Branco. I come from Lisbon, from Portugal. And I'm a medical oncologist, just a young medical oncologist, just finished my graduation in March. And I work in Lisbon Cancer Center. My main interests are breast cancer, melanoma, and also some rare tumors as neuroendocrine tumors and germ cells tumors. And so my experience in clinical practice is not that big in uveal melanoma, as we are not a reference center. So as a medical oncologist, I saw very few uveal melanoma metastatic cancer patients. So, and I recently joined Cure 2020 to this project where I work with Guilherme and Constanza. So I accept this invitation with a great pleasure, and it's an honor for me to be here today with you to share some basic concepts of cells, DNA, genes. So I also appreciate your list that helped me a lot to go through the agenda, and I will try to make it like the more pictures I can to represent all these concepts for you to make it easier. So the agenda, I will start to talk a little bit about genetics and genomics, and I will compare it as our library. So now you'll see later how can we compare this with a library. So our coding, passing through these terms of cell, nucleus, chromosomes, genes, DNA, and what does this mean? What it is invisible and visible for us, so, and talk a little bit about genotype and phenotype and what it is and the transition between the two. What is transcription and translation, all these terms about RNA and proteins. What it is a mutation and the types of mutation, the consequences of mutations, and how is this information activated. So the intracellular signaling and the predisposition to have cancer due to mutations in the genetic code. So, and after that, I will pass through the immune recognition, and I will give the example of the policeman and the thief, and you will see how will it match with this process, and what is an antigen, what is an antibody, the witnesses that see this process, which are called the presenting cells, the T cells and the B cells, what are their roles, and talk a little bit how immunotherapy can act in this setting. And at the end, I will just give two short examples of projects, ongoing projects within the Cure 2020. So starting from the eye to the cells, Guilherme gave today's morning a presentation about the anatomy of the eye. So beautifully described all the structures, and I just want to make the parallel to this, so for you to understand where the cells are and that they act as a unit, a functionary unit within the cell, which is an organ from the visual system. So if you imagine you are in a library, so you enter the library and you see a bookcase. This bookcase in the visual system is an organ, which is the eye. This bookcase has many shelves, and this shelf of retina have some cells, which are the books. So the cell, which is the book, has the information, has a message. So in the same shelf, you can have many books equal to each other, books that are different, with different messages, so with different roles. Within this book, you have a core, which is composed by chapters, and these chapters are in the nucleus of the cell, which are the chromosomes. The chromosomes, if you imagine like a landline telephone, and the line that connects to the telephone, like a spiral, are rolled all together in forming this chromosome, and are composed by genes. So the gene is the word. And the gene is composed by DNA, which are the letters. So this is the analogy to the library. So how does this start? And some of you might recall this from the school. So, and it's a common sense concept. So we receive half of our genetic information from our mother, and half of the genetic information from our father. And this genetic information is in the nuclei of the egg and the sperm. Okay, and when they join together, they create this process of fertilization and generate a cell with a full human karyotype, which is composed by 23 pairs of chromosomes. So each pair has the same genes, but one gene comes from the mother and one gene comes from the father. So it's like a copy of the same genetic information in two chromosomes, but from different origins. And one of the 23 pairs is the XY pair, which is the pair that defines the gender. So if we receive an X chromosome from our father, we will generate a woman. If it is a Y coming in the sperm, it generates a man. I will just go through these two examples of how the genotype can influence the phenotype. So imagine this example where you have two genes. This is a simplification of a process of the color of the eye. So imagine these two genes, the gene A and the gene B. So each gene has a copy in each pair of the chromosome. So the big A or the small a, and the big B or the small b are the examples of the possibilities of each gene. Okay, if you receive from and you will receive each copy from each of the parents. The big ones, the big A, the capital letters, the capital A and the capital B will give you a dark color of the eye. And the small a and the small b will give you the light color. So, and if you receive, for instance, from your mother and from your father a small a and a small b, you'll have a blue eye. But if you receive a capital A or a capital B from your father or from your mother, you'll have a higher probability of having a dark eye. Okay, so this is how it translates to the phenotype. And this also allows us to introduce some concepts of dominance and recessive. So in this case, the capital A might be dominant because, as you can see in the table, the probability of having a dark eye is higher than having a light-colored eye. So you have to have all the 4 copies of these 2 genes with a small a and a small b to have a blue eye. Another example is the example of color blindness, the most prevalent color blindness, which is more common in men than in women. And how this is possible and why is the reason is because this gene is located in the chromosome X. So the chromosome Y doesn't have it. And the reason is because for the disease to express in the phenotype, you have to be either double X and have this alteration in both chromosomes, because if you have a normal gene in the other pair of the X pair, you will not express the disease. So, and if you are a male, as you just have 1 copy in the X chromosome, you will automatically express the disease if you have this altered gene in your X copy. So this is why it is more prevalent in men, because if you receive the altered gene in the X from your mother, that could be a carrier, can have no disease because it has a normal gene in the other X chromosome. So now about transcription and translation. coming back to the concept of word, if you have your word, which is the gene in DNA information, you want to, like for instance, when I was preparing these slides, I had like genetic handwritten in a piece of paper, and I want to put this genetic in a PowerPoint slide. I transcripted this information for another form of transmit this word. This is what's happening between DNA and RNA. So it's the same information, but in a different kind of transcription. So you are transcribing the same word in another form of information, of messaging. And when you translate, it's like you are translating this word for another language. So you are passing from RNA to protein. So you're using another language for this information that comes from the nucleus to a protein. So when you have a normal gene, you will express normal protein. When you have a broken gene or an altered letter or an altered DNA in this mutated gene, you will either produce an abnormal protein with gain or loss of function, or you will produce no protein. If this word just lost the sense, you cannot translate it to another language. So, and then an example of the types of mutations is very well depicted in this image. So, if you come from the word beast and you change the first letter to an F, you have feast. And if you insert an R, you have breast. So, if you change the order, for example, you have beats. So, the mutation and the order of the pairs of the DNA will change the meaning of the word that you are translating at the end. So, in cancer, the mutations will alter this normal cell cycle division, which is a process that is always occurring in our bodies and is regulated by two types, generally speaking, by two types of genes. These proto-oncogenes that act like a gas in a car, allowing the cell to accelerate in this division process, or tumor suppressor genes that act as brakes, so allows the cell to slow down the division process. If you have an alteration, so a mutation in one of these genes, if it is a proto-oncogene that transforms to an oncogene, you will have an excessive acceleration. You will generate cancer through high division process and generation of a tumor. On the other hand, if you lose the brakes, you will have the same result. So if your cell division becomes faster and your body cannot break this division process, you will also lead to cancer. Another aspect which is also important to differentiate is the concept of germline and somatic mutations. So the germline mutations are inherited. So the gameta that generate the full DNA code are already mutated. So all the cells in the human body will have this alteration. So it is inherited by the sperm or the egg. And the gametes from this body will also be altered. Okay, and regarding somatic mutations, there is no genetic alteration at the beginning, but there is an external factor that influences and generates this mutation, which does not affect all the cells of the body. Okay. So how this can impact in the function of a normal cell? So a normal cell, usually, and this is an example of an intracellular signal, so there is an external stimuli that binds to a receptor in the membrane of a cell and generates a signal. And for this signal, it's very important to have these proteins. So these proteins are part of this of the transmission of this signal from the cell to the inside the cell, and also the nuclei. So the nucleus can be and the transcription and the translation are generated and influenced by this signal that comes from the cell membrane through all these processes within the cell. So if you have an alteration in one of these proteins, this process of signal transmission will be altered. And it is a point of research in many tumors, the way that the cell is being divided in a faster speed due to an alteration in this intracellular signal. So if there is a gain of function in one of these proteins in this chain of signals, the cell will proliferate, for example, due to an intracellular signal to stimulate the cell division. And now going to this genetic predisposition concept, and this is a well-known example of it in breast cancer and ovarian cancer, where BRCA1 and BRCA2 genes are related with a higher predisposition to develop breast and ovarian cancer. We can see here that when the mutation is present, the probability of developing cancer is higher. So it is not 100%, and this makes us talk about another concept which is penetration of the disease. So having the mutation, it's not the same that you will develop the cancer, and this is influenced by many factors. So if a person is diagnosed with this mutation, we can offer a counseling, so a screening adapted to the risk of developing this cancer, but it's not the same as saying you will have this cancer. It's not like that. And as you can see, one gene is related with a higher penetrance than the other. And this specific gene is involved in the repair of the DNA damage. So if it is a gene that is involved in repair a change in the DNA, and if it loses the function, or if the cell is exposed to an external stimuli that damages the DNA, and the cell is not able to repair it, it will generate a change, and it will generate cancer. So this is the process in this specific case. So there is also a predisposition, a gene that gives some predisposition to develop cancer in uveal melanoma, which is BAP1 mutation. And this BAP1 is a BRCA1 associated protein that has a function which is a deubiquitination function. So what it is deubiquitination? When you tag a protein with a message to degrade this protein, you put like an ubiquitin molecule in the protein. So it's like a signal to degrade this protein. And this BAP1 protein, it's a protein that it is involved in remove these tags. So it's like avoiding this tag to be degraded. So, and the process and the existence of this mutation will increase the risk of developing mutations. And there are also other driver mutations reported, which are GNAC and GNA11, which are driver mutations at the beginning. So 46, and the range of reporting of these mutations range from 46 to 88% of the somatic mutations in uveal melanomas. So now from genes to the immune system, and we will talk a little bit about the immune system tomorrow and the organs involved. And now we are going to the cells, which is the bottom part of this slide. The immune recognition is very important to understand what it is, an antigen and an antibody. So an antigen is generally a strange molecule that it is recognized by the the immune system, and an antibody is like a recognition process of this antigen, which is generated by immune cells. And it is important here to introduce this term of major histocompatibility complex, which is a complex which is related to the recognition of the self cells. So normal cells from our body will be recognized by our immune system as being from our own body through this complex, okay? So if we have this self-marker of the MHC in our own cells, the immune system will recognize as normal cells. Otherwise, if it has an antigen that is strange for our immune system, this immune system will recognize as an external protein, and then will start the immune recognition process. So I will now establish this analogy to the theft, and the tumor cell will act like a thief who's trying to enter a house with a different key. So the different key doesn't match in the door, and will start an alarm. And this alarm will call the cells from the immune system to the place, and will be made this recognition of a strange antigen that is trying to make some damage. On the other hand, there is another possibility, that is if someone sees this process and call the 911, and there is a call for some cells to go to the place and recognize the tumor as a different, as a strange antigen. And these witnesses are the antigen-presenting cells, as the dendritic cells are one example of it. And so at the very end, we will have the T cells and the B cells that are the effectors against this process of the generation of the immune response to an antigen. And at the end, with all this process of immune recognition, it will be generated a memory. So, like a record in the police. So every time that this antigen comes again, it will be easier for the immune system to recognize this strange antigen and generate a faster response against it. So this process witnesses policemen and records. So the antigen-presenting cells as the dendritic cell, the macrophage, and the B cells that can act as antigen-presenting cells will recognize this strange body, will activate effector cells that can be T cells, and will generate this memory that can be eliminate this antigen. So it will act against and will respond against this strange stimuli, and this response will lead once again to a memory that will facilitate the next exposure. And how does immunotherapy can act in all this process? So we have these two more common classes of immunotherapy drugs that we commonly use now in cutaneous melanoma, which is CTLA-4 inhibitors and PD-1 inhibitors. And they act in different places, but in generally concepts, they do the same. So they inhibit an inhibitory stimulate to the immune system. So there is these receptors here, so CTLA-4 and PD-L1, which will inhibit the immune response. And if you give a drug like PD-1 inhibitor, we will inhibit this signaling through the tumor cell. Okay. And so this is a sort of immunotherapy which is commonly used. Another types of immunotherapy are, for example, the vaccines that will generate antibodies specific to the given antigen. So the antibody immunotherapy that we have already talked about, this blocking the inhibitory effect. And the antibody immunotherapy, when an antibody is generated in the laboratory to target a specific antigen and give either a chemotherapy conjugate to eliminate those cells that express this antigen, like tumor cells, or that can block cell signaling process that starts in that receptor, in the specific cell that expresses this receptor. And you can also have the cellular immunotherapy with the CAR T cells. There are T cells designed to attack or to target cells, cancer cells that express a given antigen. And the same for dendritic cells targeting that are aimed to activate the immune system against a specific antigen. And shortly, these two last slides just to give you two examples that within UMCure 2020 are exploring this relation between genetic alterations and immune activation. So, for instance, Professor Marc-Henri Stern, who is very interested in this area, and as you might know, produced a lot of work from Institute Curie, who published some years ago this case report of a patient that has a particularly good response to immunotherapy due to a genetic mutation that is related with a lack of capability of the cell to repair DNA damage. So if you have a DNA repair damage lack of capability, you will have more mutations in your cell and you will have higher probability to develop cancer. And this generally speaking, has been already shown to be, so the tumors that have more mutational burden, so higher amount of mutations within the tumor cells, are those who are believed to respond better to immunotherapy. So this could be a way to identify a tumor and the patient that has this tumor that can eventually respond better to the immunotherapy. And also, Professor Olivier Lanz, who also is doing a lot of work in this translation immuno-oncology, and who also gave an update last week and in Paris in our progress review meeting on the correlation between specific mutations in uveal melanoma and the recognition of T cells that can recognize cells that have these mutations in the pathogenesis of the disease. And that's all.
Fantastic, thank you.
Have you heard the first part or not at all?
So the first part of the question was that it's often said that melanoma is a disease of the melanocyte. What function does the melanocyte actually have in the eye?
So in the eye, we can find melanocytes in different structures. The ones that we found in the uvea, that are in the iris, the ciliary body, and the choroid, they might have different functions depending on where they are. For example, in the iris, they can act as a curtain to block light to enter the back of the eye in an excessive amount. In the ciliary body, they may help the other cells to produce the fluids inside of the eye that help nourish the other structures and maintain eye shape. And in the choroid, there is a great debate from where and why melanocytes arrive there. And perhaps Professor D'Amato can help me answer this part. But melanocytes tend to have also a role in image formation in the choroid, as act as blockers for the excessive amount of light that can pass through the retina itself.
Thank you. Thank you.
In a simplistic way, I think of a camera which is usually painted black inside to prevent too much light bouncing around and causing blurred images.
So maybe starting from this one, how can cancer cells, how are cancer cells able to hide? And this is, for instance, one example that I gave in that the process of immunotherapy in cancer, so how a treatment that inhibits the inhibitory stimuli. This is one example of how a cancer cell, through these receptors in the cell membrane, can inactivate the recognition process and then hide from the immune system, for example. Other ways that the tumor can resist to the treatment is through developing mechanisms of resistance to the treatment that we give as a systemic treatment, like, for instance, chemotherapy. There are a lot of different kind of resistance process already described, as, for instance, some channels that can open the membrane to eliminate the chemotherapy to the outer part of the cell, and then the cancer cell can resist and survive to the treatment. And so these are two examples of how cancer cells are intelligent to avoid either the treatments that we give through a systemic treatment or hiding from the immune system itself. So, is there any genetic counseling for pregnancy with ocular melanoma? As I know, I don't think so. For at least in Portugal, we don't do it in our center. But I don't know if you have something to.
So, uveal melanoma does not tend to be passed on from a parent to a child. So, these alterations that Diogo explained, these mutations, they happen in the patient itself. So there is a rare form of uveal melanoma that passes on from family member to family member, but it's associated with other problems as well. So it's not the regular uveal melanoma patient that has these types of mutations.
Yeah. So this BEP1 predisposition syndrome, which is it is much more rare than the BRCA1 and BRCA2 example that I gave you. So probably it's more difficult to predict what is the clinical impact of this alteration in genetic counseling, because we don't know as good as we know the penetrance of this alteration to generate ocular melanoma as we know for breast cancer, for instance. So this is why it's difficult to counseling with this alteration.